Welcome to Medical Lab Science Quick Review. We will highlight chronic lymphocytic leukemia, pro-lymphocytic leukemia, and hairy cell leukemia, followed by questions and answers revolving around topics in the AACP Board of Certification Study Guide. The World Health Organization classifies the mature lymphoid neoplasms as a diverse collection of chronic blood and lymphoid diseases that involve transformation of lymphoid cells at various stages of development. In the clinical lab, diagnostic tests such as peripheral blood and bone marrow morphology, histochemistry, flow cytometry, and molecular testing help identify and sort out this complex classification. So let's get started on CLL and then later on chronic prolymphocytic leukemia and hairy cell leukemia. The people with CLL are typically elderly in their 70s. Some are asymptomatic who comes in for a routine clinical checkup, while others may complain of symptoms of a fulminant illness such as fever, night sweats, weight loss, lymphadenopathy, gastrointestinal or neurological symptoms. Right away, we notice leukocytosis and relative lymphocytosis because we see that these values are higher than the AECP reference ranges. And we know how to calculate absolute values. And we see that our calculation for absolute lymph count is high. On the CBC histogram report, flags prompt us to do a peripheral smear exam. At high power magnification, we see a monotonous sea of mature limbs with many smudge cells. Smudge cells are artifacts of fragile, mature limbs. They are breaking up as we or the instrument makes the peripheral blood smear slide. By the way, there was a question about smudge cells in our ACP study guide. FYI, for a more accurate diff, mixing albumin with a few drops may eliminate most of the smudge cells, although usually it makes no difference. You don't see blasts, but in some cases of CLL, you might find a few immature limbs, the prolimbs. You don't see eosinophilia or basophilia or any of the myeloid precursors. We'll use the oil immersion for a close up of a mature lymph and use it to distinguish it from other normal and abnormal leukocytes. The neoplasm in CLL involves a monoclonal B cell lymphocytic malignancy in the bone marrow and blood, a key distinction from small cell lymphoma, which occurs in lymphoid tissue. Did you know that CLL and small cell lymphoma represent different clinical manifestations of the same disease? One of the complications of CLL is autoimmune hemolytic anemia, where you have autoantibodies and complement with a membrane attack unit affecting hemolysis. Chronic prolymphocytic leukemia is a rare and more aggressive leukemia that involves monoclonality of either T or B cells. Prolymphs appear larger with nucleoli, more open chromatin pattern that may appear cerebriform, and ample blue cytoplasm. You can call them immature lymphocytes if you're not sure of the identification between lymphoblasts and prolymphocytes. Hairy cells are described as abundant, pale staining cytoplasm with hair like projections around the circumference of the cell with oval nuclei and relatively fine chromatin. The one cytochemistry stain to remember in hairy cell leukemia is tartrate resistant acid phosphatase, TRAP. Hairy cells have acid phosphatase enzymes which are resistant to tartrate, so after a counterstain, we see them as the light pink version of mature limbs. The flow cytometry instrument is a very sophisticated diagnostic instrument used by medical lab and clinical lab scientists after they prepare and test specimens and gauge cell populations in the computer for the pathologist to evaluate and make a diagnosis. Flow cytometry is useful for blood, body fluid, bone marrow, and lymphoid tissue cell suspensions. Flow cytometry uses a highly specialized flow test system for immunophenotypes represented by CD for cluster designations, which are colorful fluorescent signals lit by a laser beam. Gated fluorescent signals represent normal and abnormal white blood cell populations tagged with reagent fluorescent conjugated antibodies. This is a somewhat simplified version of a long, exhaustive list that veteran flow cytometry specialists are used to in their fascinating field of work, but for the generalists, note the highlights. Think of CD in the teens and 20s as of B cell origin, so not surprisingly, we see a lot of B cell markers. CD5 plus represents T cells. Hey, wait a sec, we know that SCL and CLL is the same disease with the B cells, yes, but aberrant expression of T cells are also involved, as noted in the text and AECP study guide. CD5 and CD23 are characteristic of CLL and small cell lymphoma. Please note that in hairy cell leukemia, it is known to be CD11C positive and CD103. And now for some Q&A that is similar to the ACP Board Certification Study Guide. Thank you for your time, attention, and your support in helping those afflicted with cancer.